Hello and welcome to our last topic of the year. This is going to be the nervous system. Just like all of our other body systems, we're going to follow through in the same order where we're going to learn what the system is, and then we're going to take a look at how it works, and then maybe what happens when things go wrong. A few things that are different about the nervous system is that with every other body system, if something goes wrong, you can kind of make sense of it, right? So if a stomach has some issues, you get some indigestion, or either way, digestive system, if there's an issue, food either comes out in one direction or it comes out in the other direction. Respiratory system, you get a breathing disorder. Something happens with your ability to breathe. The nervous system is a little bit different because this is your brain we're talking about. And so your body actually tries to trick you. It doesn't tell you necessarily when things go wrong. It tries to make sense of what's happening. So it's a very interesting one. It's a really fun one. And historically, I find that this is the one that people really enjoy. We're going to break it into a couple of parts, starting off with part one. Here is an introduction. And sorry, this first section of notes, these green notes, are going to be an introduction. We're going to take a look at a separate set of notes for senses, and then we'll do a couple of activities in class where we can try and test out what happens to our nervous system when things go wrong, but we catch it. We don't let our brain just trick us. We catch our brain tricking us, which is always kind of interesting as well. Now, I should also note, if you're checking this out on the Google Classroom, you might notice that the notes on the Classroom include slides that are skipped during this presentation. Just for the sake of time, we've removed some information, but I promise those slides that we're missing aren't missing any information. So if you want to read through those, you're more than welcome to get that additional information there. But we're going to cut a little bit of that out for our section. Starting off, when we talk about the nervous system, we have to understand what the nervous system is. And it's got two divisions. You got the central nervous system, or the CNS, which includes your brain and spine. Or we can take a look at your peripheral nervous system, which is the PNS, which is everything else that's connected to that system. So your brain and your spine comes down the middle of your body, and then your peripheral nervous system is everything else that's connected there. All those nerves and fibers that connect to that brain and spinal cord. Inside of this nervous system, you're going to have two different kinds of cells we're looking at. More commonly, we're going to really look at the neurons, which are the actual functional unit. Those are the nerve cells. We've also got neuroglia, which are going to support and take care of these neurons for us and prevent them from getting damaged. But we're not going to spend as much time talking about those. We're mostly going to be talking about those neurons for our course. So if we take a look at the nervous system, we can see that here's a spinal cord, just as an example. There's different kinds of nerve cells that you can get. Here on the top, I've got what's called a sensory neuron. This is for any sensation that you're going to be experiencing. The nerve cells are very long, string-like structure, and it's got a cell body with the nucleus sticking out of the cell itself. A motor neuron is slightly different. It goes from your central nervous system to an effector organ. It could be a muscle, it could be a gland of some sort, but a motor neuron is where you discharge that electrical impulse. That has a cell body inside of the cell. Lastly, we have other types of nerve cells that we're going to look at in just a second here, which are kind of in between. They're the intermediary neurons that we look at. So just to have them as a note, you have three different classes. The sensory neurons are the ones that give you sensations. So anytime you sense something, and don't get trapped into that whole idea of five senses. You have way more than five. You have anywhere between 180 to 215 senses or something crazy like that. Things like sense of balance, sense of proprioception, sense of temperature. You got hunger, you've got thirst, like all these other weird sensations that you get. Those are all full sensations. If you really break it down, a sensation is just a really detailed touch of some sort. But if you want to take them into separate categories, you have hundreds of these sensations. You use sensory neurons to get those signals. They send those messages to the CNS. And before it gets to the central nervous system, it has to go through what's called an interneuron, which is kind of in between a sensory and a motor neuron. An interneuron is going to decide whether to send a message right to your brain, so into your central nervous system. The benefit of that is your brain has the processing power to make a more complex decision if necessary from that sensation. But in some situations, that interneuron will send the message directly to a motor neuron to cause an effect where thinking about a situation might not be as valuable. For example, if you're cooking in the kitchen and then you put your hand on the stovetop and you sense that it is hot, That is not information that needs to go to your brain. Your brain doesn't have to tell you that this is hot, you're going to burn yourself. Instead, your interneurons are going to send the muscles directly to your arm fibers using the motor neurons, and that's going to cause your arm to twitch away from your stovetop so you don't end up burning yourself. In the case of, let's say, maybe you're playing a sports game and you got to make a decision on, let's say you're playing soccer and you got a player to your left and a player to your right. And so the sensation is you know where they are, you know where your comfort is, you recognize that you have to pass somebody the ball. Who are you going to pass to? What decision are you going to make? That's not something that you can rely on impulse. You can't rely on the interneuron to send that signal. Instead, that signal has to go to your brain. Your brain's going to calculate what are the odds of passing in this direction, what are the odds of passing in this direction, and then depending on how good of a player you are, you might have more processes going as well. That's going to help you determine where to send that signal. So it takes a little bit longer, but you get a much more detailed, complex, thought out situation if we go past the interneuron into the central nervous system. Neurons themselves, they have a specific shape and body to them. So even though they all look slightly different, they have some similarities. For one, you're going to always find the cell body, which is going to contain the organelles, the nucleus, all that jazz. We've also got dendrites. Dendrites are the starting point of any of these neurons. They have a whole bunch of little feelies, kind of like roots on a tree or the branches of a tree, even, and they're going to collect that electrical signal and bring it into the neuron. On the opposite end, you have the actual body, the length of that neuron itself. That's called an axon. And axons are what are going to carry that electrical impulse through the neuron. I'll show you a picture of what that looks like here. Just up at the very top, let's say, for example, you're touching something. So this is a skin, cube of skin. I don't know how else to describe that. You have dendrites in your skin, which are going to receive the impulse when you touch something. So when you touch something, that pressure change in your finger is going to cause a reaction here in your dendrites. That is going to cause the electrical impulse to move through this axon. This axon is going to take that charge, send it to the axon terminals, which are going to terminate over various other dendrites, which are from other neurons. In this example, they're going to interneurons, which will be in your spine. Before I go any further, I should also point out you're not typically going to go directly from your skin to your interneurons. You're probably going to go through your skin and then multiple axons first, and then you're going to enter an interneuron, which is going to decide whether to send a signal to your brain. So let's say you're touching, I don't know, you're touching your skin. Don't really have to think about that so much. It doesn't have to be a fast response. We're not going to send that signal back to your motor neurons. We can send that signal to your brain and decide what to do. If instead you're touching something that's very hot and you don't want to burn your hand, that signal is going to go through the interneuron into what's called a motor neuron now, which operates the same way, receives a signal from a dendrite, goes through the axon, but this time it terminates onto some sort of effector. In this case, it's on the muscles in your arm that's going to cause you to twitch away from whatever was causing that sensation. If we continue on with the structure, you might have noticed there's a layer that seems to be covering all of these neurons. This is called the myelin sheath. Just like an electrical cable, this is going to be some sort of insulation, which is going to prevent your neurons from losing that electrical signal. This insulation comes from what's called a Schwann cell, which surrounds itself on your neuron. Again, it gives you that insulation. The space in between the myelin sheath is going to be called the nodes of Ranvier, and that's where your charge is going to be jumping in between. You have some of this myelin sheath around various parts of your neural network. In your brain, for example, the exterior portion is covered in gray matter, which does not have the myelin sheath, so you lose a lot of those signals much more easily. So that's like when you get an idea, it's on the tip of your tongue, like you're thinking of something, but you can't quite get it out. It's because your brain knows the answer. It's trying to send the signal to your body or to other parts of your brain so you can make that response. But unfortunately, you're unable to get it there because you don't have that insulation to keep the signal there. On the flip side, if you get a deeper thought, something that is further in your brain, for instance, things like anxiety and pain are often remembered in a deeper part of your brain. You have white matter to insulate those electrical impulses. So you tend to remember those things. Those things stay much more easily in your brain. If you're really good at using your brain, you can get some other useful thoughts in this part of your brain, so you can you keep yourself remembering important details. Your spinal cord is similar, but kind of the opposite. Similar in the sense that it has both gray and white matter, but the gray matter is going to be in the middle this time. The white matter is going to be in the outer regions of your spine. And just to see what that looks like, in the bottom right here, I've got a Schwann cell. Again, this is made in the PNS, and it essentially just wraps itself around your axon And it does this in multiple layers, so you get all these different myelin sheaths. And this empty space in between the sheath is called the node of Ranvier. So that electrical impulse is going to start and it's going to bounce between these nodes all the way down one of those axons. Typically, they move in one direction, just depends on whether you have a sensory, which is going to go towards your central nervous system, or if you have a motor neuron, which is going to go away from your central nervous system. The impulse itself, this is a more difficult idea to understand. It is electronic, so we should be able to calculate what that charge is going to be. So when your nerves are not firing, they have what's called a resting potential, and we can measure that with some electrodes to be a negative 65 millivolt electrical potential. This is because, and again, this is where things get a little bit weird because we're talking a little bit about chemistry here, where we have to understand some biochem to understand this resting impulse. So if you don't have a background in chemistry, it's okay, but we're gonna use a periodic table to help us out a little bit. On the periodic table of elements, there's the way that the table is designed is so that you can read it in whichever direction you want. So you can read it up and down, you can read it left to right, you can read it diagonally. Depending on which way you read it, you get different information off that table. If we read our table from top going downwards, so if we start at the very top and we read downwards, this is telling us the size of the elements on the periodic table. So at the very top, you have the smallest elements. The lower you go, the larger the elements actually get. This resting potential is due to two elements in particular: sodium, which is number 11 on your periodic table, and potassium, which is number 19. Both of these are on the far left, the alkali earth metals, that alkali metals category at the very, very start of that periodic table. Sodium is one place above potassium. So that means potassium is significantly larger than sodium is going to be. Those sodium ions are going to be more plentiful outside of an axon, whereas potassium is going to be more plentiful inside of the axon. Now, there's a little bit of a misconception here, because both sodium and potassium are positively charged. Why the minus 65? This is because potassium, because it's larger, it takes up more space. So there's more repulsion between various potassium ions inside of that axon. That empty space in between that keeps the structure together are going to be negative ions, likely a chloride. It could be maybe a fluoride or other negative electron charges. Those are going to give it the minus 65. There aren't too many of those negative charges on the outside of the axon. Now, what ends up happening is we have the ability, using what's called a sodium-potassium pump, to move potassium out and move sodium in. If we move sodium in, we're creating more space because sodium is significantly smaller. We're not changing the charge, though, because both of them have a positive 1 charge. So that's going to help us move that negative 65 down throughout one of our axons. If we do ever get a chance to do that, we call this an action potential. And this is when this polarity changes all the way across. At minus 65, if it's moving, it means there must be some sort of change happening in that axon itself. If you do ever pass what's called a threshold stimulus, you get to change that voltage at a certain point, then you actually get an action potential. If you don't hit that threshold stimulus, you will not have the signal moving through your axon. This is going to be called a depolarization. We'll give you a note of that in just a second here. And this is going to be due to what are called channel proteins. Channel proteins we talked about way in our first unit. We have one for sodium and one for potassium, and they're going to dictate what goes in and what comes out of that neuron. So let's walk through it to try to understand it a bit better. First off, your sodium gates are going to open. When sodium gates open, that allows sodium to enter that axon. The sodium is going to try and cancel out the chloride that was there. So the positive sodium is now canceling out the chloride that's on the inside of that axon. As a result, if we cancel out the negative charges and you still have potassium, you just have positive charges now. So that potential changes from minus 65 to positive 40 millivolts. Now you've done something called depolarization. You've changed the charge, and now that charge should wave through that axon. It should move across that axon, and that's how you move the charge through one of these axons or through one of your neurons. At the end of this, your sodium gates are going to close, and then your potassium gates are going to open. Because of the intense repulsion of all these positive charges, potassium easily, even though it's large, is going to be kicked out of that axon. So now it's leaving that axon. You're losing positive charges. And this is going to happen until things balance back out to a minus 65. So essentially, what's happened is the sodium and potassium have changed positions, and that's going to bring things back to minus 65. This is called repolarization. The issue we have now, though, is we have two different charges, but our sodium potassiums are in the wrong spots. We're going to need some help to actually fix that later on, but that's more of a biochem topic. So we're not really going to get into that, but we're going to talk about neurotransmitters in just a second here. They're going to be involved in moving the sodium back out and the potassium back in. To put everything into perspective, all of this is happening within 1 millisecond, and that's for every single effector in your body. So if you're watching this while sitting down, you've got muscles in your abdominal cavities, your abs, your back muscles that keep you sitting upright. You have your diaphragm helping you breathe, your cardiac muscles helping you pump blood throughout your body. Your brain is thinking, you're listening, so you have auditory signals, receptors in your ears that are picking up some sort of message. You're not tipping over one way or another, so you've got some balance receptors. Your world ideally isn't spinning upside down, so you've got some proprioception. What I'm getting at is you are firing a ton of these polarizations. You're depolarizing so many of these neurons every single moment that you exist, because this is happening every millisecond if anything is happening in your body. And from the moment you're born, or even from before when you're born, until the moment you die, including even moments after you die, these are still happening. There are still things that are functioning in your body. Even when you sleep, you're still firing neurons. So this is always, always happening. Just so that we can see it visually, you can see that you have the sodium, which is positive on the outside. You have the negative charges on the inside, so they try to cancel each other out. If we zoom in on this piece here, so this yellow tube is an axon. If we zoom in on that, you can see the different gates, green for sodium, yellow for the potassium. First, the sodium is going to come in and enter the cell to balance out the green and the yellow. And then afterwards, the yellow is going to go through these gates and come back outside. That's the whole process. And we're measuring this using what's called an oscilloscope. You can see someone using one on the end over here. So what exactly happens? Axon is carrying that electrical signal. That signal is going to get at an axon terminal. It should now connect to some sort of dendrite. This is going to happen across the space between the axon terminal and the dendrite of another neuron. They don't actually touch typically. There's a space in between, and that space is what's called the synapse. That's that junction in between the terminal and that dendrite of another neuron. As a result, you have a couple of vocab terms to worry about. The space where there is absolutely no contact is called the synaptic cleft. If we're talking about the first neuron, we call that the presynaptic membrane. If we're talking about the second neuron, it's the postsynaptic membrane. Neurotransmitters are going to be involved in moving one of those charges from one neuron at the axon terminal to the dendrite of the postsynaptic membrane. Neurotransmitters are simply chemicals that are going to help transport that charge, that chemical charge, across that synaptic cleft. It does so using a separate channel system. In this case, it uses a calcium channel lock. And the calcium channel, if you have calcium in your body, is going to use synaptic vessels to move that charge into the next dendrite. That way you can continue moving that charge all the way throughout your body. These charges can either be excitatory, which helps you increase the odds of an action potential, or inhibitory to prevent that from actually happening. An example of inhibitory is when you have a headache, for example, you are sending pain signals in your brain. So your sensation is pain. When you take an Advil or a Tylenol, those are all inhibitory medications. They're blocking that pain. So you still experience the pain, but you don't actually perceive it. So your brain is still hurting, but you're not experiencing it because you're inhibiting that sensation. Excitatory is the opposite. Excitatory is when you do something and it's something that you take that increases the likelihood of an effector actually happening. So example of an excitatory action is you're sitting on the couch for a while and you just want to get up. You've just been lazy for too long. You want to get up. So you're getting all these signals from various parts of your body. Your leg muscles want to wake up. Your brain wants you to wake up. You kind of feel like you want to go for a jog. You want to do something. All of these signals, these thoughts are sending impulses to various parts of your body. They're adding together. They're exciting you. These are going to cause you to actually have an action potential. And that might cause your leg muscles to clench. And that allows you to stand up and then you can get up off the couch. Here you can see Axon terminals, they end up at the end of some dendrites, and then in the space in between, you have a synaptic cleft, and then you have the neurotransmitters that can cross over that synaptic cleft. Now, what happens when you have multiple axon terminals onto an individual dendrite? Because dendrites have all these little feely finger pieces like the roots on a tree. So you can have multiple axons connecting. You get what's called integration. Integration, for the sake of grade 11 biology, we're going to say that that's just the adding or subtracting of electrical signals. So if you have excitatory signals, they add together. If you have inhibitory signals, they cancel out excitatory signals. That is simply integration. You're just adding those signals up together. If you have excitatory and enough excitatory signals, good chance that you're going to hit that action potential. If you get more inhibitory, you're probably not going to hit that action potential. And if you get both, they kind of balance each other out. Depends on which one is stronger. Here you can see you have one neuron, which is sending you an excitatory signal. That's the red one. The blue one is sending you an inhibitory signal. And the black one is what that new neuron is actually going to experience, something in between. In this case, you don't have enough excitation, so you're not going to hit the threshold. And because you don't hit the threshold, you don't actually end up firing the next neuron. So you need more excitatory signals. If you were to hit this threshold, though, then you would actually have that impulse release. The way that your system organizes things, the central nervous system is going to communicate both ways, through motor and through its somatosensory system, so your sensory network. It's going to go through cranial nerves or spinal nerves, and then through sensory or motor fibers, and then things change up a bit. Only your motor fibers can cause you to actually move and do things. Only your sensory fibers can send signals back to that central nervous system. So arrows don't go in both directions. And that is going to bring us to part 2, where we're going to talk about the brain. In our class, we went beyond part 2 to talk about and introduce the brain a little bit, but we're going to pause here for the video section, and we'll do part 2 in the next section. We'll talk about and label different parts of the brain and try to understand why and where some of these thoughts are coming from. For the time being, though, we're going to cut it here, and we'll maybe take this up on our next video. Anyways, hope you're having a good one, and we'll catch you later.